We have a number of other talks each Wednesday, 12 to 12:50, right here in Sites 209, and also available by way of Zoom. So today, it's our pleasure to introduce Dr. Ramalho from the WCU Biology Department. She will be telling us about her work and research titled Fostering and Understanding Diversity Within the Ant Microbiome. Thank you.
Thank you, John. Thank you everyone for being here. As Joseph already mentioned, I'm a biology professor in the department here in Westchester, and it's always a huge pleasure to talk with you all. Let's see if that will work. It is working. So my seminar today, I will first talking how insects are awesome, and then we're gonna focus a little bit about introductions of symbiotic interaction and how this is really important for the diversity of insects that we can see on Earth today. And then finally, I will focus specifically my favorite types of insects, with ants, and what the importance of ants for ecosystem. And finally, we're going to move the talk for how the host ecological factor and the host phylogeny can help to shape the microbiome community or the microbiome of insects. And finally, I want to use the last minutes of my talk to talk about the importance of diversity and inclusion in science. You're going to see during my talk that I have really pleasure to show results of 3 senior students from Westchester that will graduate in May. So I hope that you get excited the same way that I do. I'm excited, but also sad, like when they leave, it's pretty sad, right? But I'm excited that they go outside, and Ethan is here, so you're gonna see some of Ethan's results today. So bear with me. So besides bacteria, ants are the majority of life on Earth. And I study the insects and the interactions with bacteria and other microorganisms as well, but main bacteria. However, it's not, I hope that this is not new for you, but there are several decades of studies that have been demonstrating for the last decades that insects are reducing, their populations are declining. And finally, after a lot of years of publications, we entomologists finally reached like the higher media, publishing journals like New York Times, saying that it's what's happening in the last couple of decades. It's called now insect apocalypse. So there are several reasons why the insects are declining, and climate change is definitely one of them. But besides the climate change, and one of the impacts of climate change is also due because of us, humans. So I'm from Brazil. I was not born in Amazon, but Amazon is a huge impact, not just for Brazilians, but for the entire ecosystem. I'm bringing Amazon an example, but what is happening in Amazon is actually happening in several biome diversity all over the world. So Amazon's, it's great. Everyone knows that Amazon has a huge diversity for not just insects, but everything, including bacteria. And due to humans' activities, we are destroying these biomes and destroying every single biome on Earth. And there are several studies, and these studies are focused not in Amazon, but there are several studies that are demonstrating that even just a few years ago, we still continually devasting, destroying natures like Amazon. So humans, even though we know now, we have the knowledge that that's really important and crucial for our survival, in very anthropocentric kind of view, our survival, even though we still continue doing the same. So with that, we are not just destroying the diversity of that biome because everything is connected on Earth, but we are destroying everything, including ourselves in this process. And we always keeping thinking when we saw like the destructions of like important biomes that there's like very charismatic furry animals that are being destroyed over the process, right? Together. So that's the case of Amazon of this monkey, that's beautiful panther as well. But for insects specifically, we are destroying them and making them extinct before we actually discover that they are there. So these are still, everything is being tracked, including the little ones that are really hard to find and quantify. So in case specifically of these ants, this is one of the most important ants on Earth. It was collected just once in Africa. One ant, not one colony, one single ant was collected. And I will show you why these ants are so important, but just like, stay tuned. It is literally the basal lineage of ants for all the ants that live on Earth. And we collect just once in Amazon. And a few years ago, I was there and I was looking to the ground. At the time, did not collect the entire colony. I need to come back there. So stay tuned. Maybe we'll see in the news the Westchester professor found the ant lineage important for all the world, but so far, not success. But I hope that you now understand how important biodiversity it is for not just the world itself, but also for us. And insects are quite diverse. They have different shapes, different colors. They can get different reach. They can influence the environment of the ecosystems as different ways. And several studies have been demonstrating that the insects are able to do that because they engage in several symbiotic interaction, not just with other insects, but also with some microbes and plants as well. So symbiotic interaction, they are quite diverse and they are quite challenged to understand and to study because you need just not to study one species or one group of animals. You need to actually implement several different unique organisms and see how they tie together, how do they engage with each other. So this is, if you want to study this, great. Now we have technology and we are in a good shape to understand more about them, but it's really challenging, also fun. Like I'm young in my career, I like to think that, even that I'm turning 40 this year. So I hope that I will be here for the next 30 years in Westchester studying that. So if you are, I saw some of the students here from my library, so if you, the good news, we have tons of work to do. But let me show you how complex these interactions can be and how challenged that can be. So example here, I have a bug in a bug in a bug. So we have this malibug here that you're seeing this screen that has a diet that is deficient in amino acids. What do I mean by that? Just by going outside this malibug and eat all the food that they find, it's not enough where they get all the nutrition that they need. So these malibug over the years acquire a bacteria, here highlighted in blue, they call Tremblia. So Tremblia, fun fact about this bacteria. So these bacteria have one of the smallest genomes known so far. What we mean by that is now Tremblia is not able to do this job alone anymore. So what Tremblia did? Tremblia acquired another endosymbionts that you're here highlighted in red called Moranella. So another fun fact about now Moranella was named after a woman in science. So Dr. Nancy Moore. So a dream come true. Maybe one day you'll have my name in one of bacteria or ants, right? Whatever comes first, I'm happy with that. But look how crazy it is, this interaction. Moranella lives inside of Tremblia, lives inside of the malibug, and that's a perfect example of a bug in a bug in a bug. Could be very complex and challenging to study. But what causes this symbiotic change? How can they arise in first place, in evolutionary speaking? How can we explain all of the diversity of those interactions that we see out there? So those are the kind of questions that intrigue me and I'm really interested to study. And one of these key studies conducted by Lei and collaborators in 2008 were able to study different groups of mammals, and they demonstrated that bacteria community could be explained by the different diets and the different phylogeny of the host, the different evolutionary history of the host. So we still have tons of open questions regarding these, and when in 2008 we started to study those kind of questions with the technology available today, we thought that we would find one big truth that could explain all of the diversity of those interactions happening. What we're seeing so far, after a few decades later, it's like, yeah, it's not that quite right. So every own system has their own caveat, which again makes our challenge as a scientist to study those like complex but also fun. So what do we know so far for bacteria community specifically? We know that overall the host phylogeny, the different development stage of the host, the different diet and the different geographical location where those hosts happen, that could help us predict to influence the bacteria community associated with that. However, what about other systems besides the bacteria, the microsystem? What about all the fungi and the nematodes that live inside of those hosts? What about all the viruses that it's not even considered living organisms, right? How all of these play a role actually to the host. So I hope that you understand by now that that's a field that it is in change, and it's definitely you use that knowledge to continue understanding more about what it is having already outside in the world, not just in Amazon, but in more the natural area as well. Now we show some example of ants that we collect here. I hope that I can convince you that we have a lot of open questions, but my next goal is convince you how ants are pretty cool for answering or to study all those questions. So ants, they happen literally everywhere in the world. If there is a human, there is an ant. We have describing now more than 17,000 of species of ants. So humans, just one species that kills everything. Ants, way more. They also have quite diverse habits. They can eat everything. They can be herbivores. They can be frugivorous. They can have different shapes, different size of the colonies. So there's quite diversity associated with ants. And there's these groups of ants here, and several of those species, groups of ants, they have these interactions with bacteria communities for more than 50 million years old. That's the case of Campanotus, what you're seeing here, Campanocini ants, and this bacteria Blockmania. So these bacteria, it's really important for the host, especially in the beginning of the development stage, because they provide an nutritional upgrade, help them to process get all the nutrients that they need from the food. And with that important function, helping nutritionally, makes sense that the queen, the mom, send this bacteria yet through the egg to the next generation of ants inside of the colony. So I had a chance to study this in my PhD, and I was able to understand. So that's a queen, that's ovariole of the queen, how the eggs are made yet inside of the belly of the queen. So when the bacteria actually reach the egg yet inside of the ovariole of the queen before these legs being laid down. So there are several bacteria that happen yet in the egg, but they all use different evolutionary strategy to reach the egg. So it was really interesting to study this using molecular techniques and fancy microscope to get exactly the moment when bacteria reach the egg. Okay, so there is a famous scientist that study ants that's famous all over the world called Edward Wilson, E.O. Wilson. And so he studied ants, and he was famous to study biodiversity all over the world. And he was actually my grandfather in academia. So people who study ants are very lucky to have this famous scientist that study ants. And he wrote several books, and this is the last book that was published from him. And I was reading this book, and I was very intrigued by this sentence here. So I will try to read you, sorry, bad English when I'm trying to read, but bear with me. So for every problem in biology, there exists a species ideal for its solution. And conversely, for every species, there exists a problem which study it's ideal to solve. And I was thinking a lot about this sentence for a while, because the way that I think about microbiome in ants is actually pretty much that way as well. But my change will be for every microbiome problem in biology, there exists an ant species ideal for its solution. And conversely, for every ant species, there exists a microbiome problem, which its solution is ideal to solve. And with that mindset, I've had a chance to study sometimes just one bacteria at a time, or all the bacteria at once when you study microbiome to look like, take a look at the diversity of ants there are there. So I had a chance to already work with Campanotus, Dasyphyllum, Cephalotes. Look that crazy ants have. Colobopsis, look that face, adorable. Polyrax, spiny ants, Atta, which is liver-coated ants, Solenopsis, fire ants, everyone hate that, right? Manu Ramalho, the tiny ants that live inside of our kitchen, or at least live inside of a kitchen in Brazil. Tapinoma ants, we have in the Department of Biology all over the place. If you look at them walking the wall, it is Tapinoma. Phaedoli, the big head ants, Ecophylla. Do you know that we have a green ant? Yeah, Australia, go there. And Aphanogaster, which is the local ants here that we call it in Westchester. So it's different shapes, different colors. And before I move on with my talk, I have a lot of great pictures of ants, and this is all due because we have great ant photographer, Alex Weiss. So when I give it enough credit now, but if you saw a picture in this talk that there's no his name, that's Alex White. And you should go check his work because it is incredible. Okay, so I told you that we have ants all over the world, and so we also have scientists who study ants all over the world. However, there is places that we know more the diversity of ants and the diversity of bacteria compared to other ones. So here in this study conducted by Finn and myself, that is still in prep, we are mapping in this world where those ants were collected, where those ants have their microbiome studies. We saw that majority of like the huge chunk of the world is still not know as much in terms of ant diversity and the bacteria who lives in those ants. So they hope that this paper, when it's finalized, that we will be calling for other future studies between ants and bacteria in those places. So is anyone joining me to collect ants in Australia or Africa or a huge chunk of Asia? Yes, I saw some hands. Awesome. So there's a lot of still unknown about those interactions on nature, considering the diversity of ants and the diversity of bacteria that are still out there. And this year, finally, we, using the most up-to-date technology available to study evolution of ants, we using ultra-conserved elements. I'm not going to enter into the details, but it's a very genomic, high-skilled kind of technique. But we have the most up-to-date phylogeny to understand the evolution of the tree, evolution of ants. And remember that I told you about Marchalis, that ants that we collect just once in Amazon and no one ever collected again, literally is the first one here. So to all the diversity of ants on Earth, that's Marchalis, which is represented over here. So we now have the most up-to-date phylogeny with all of them. And what we can see when we map all those studies that focus and study bacteria and ants on the phylogeny, that we still have long lineage of ants that was never, ever looked at to see what kind of bacteria they have, what kind of holes those bacteria will have. So there is a huge groups of ants out there that we can collect and study, including ants that live in the US. So in Gordio Natural Area, that I will show the first time how do the microbiome looks like in a few minutes. So I hope that I can convince you. I can check my time because I like to speak, professor. So you need to see me speaking Portuguese, incredible. So I hope that now I can convince you that we still have tons of questions open regarding those topics. Diversity of ants, diversity of bacteria, both of them together, way more. So today I will focus on what some ecological and phylogeny factors can help us to explain both the compositions and an abundance of microbiology diversity. I will focus on bacteria for today, just for the sake of time. And I mentioned with you in the beginning, I have 2 senior students graduating that work in my lab this year, and I will show the 3 results that these 3 different students are working currently. Some of them was published last week. More they will publish soon. So I hope that you get excited the same way that I am and proud the same way that I am. But it's sad that they're leaving. So for the ecology part, I want to focus in 4 different studies. So first of all, I will focus in Daxatone, one of the incredible ants, Ada Saxons, to talk about the different environmental. In that way, that one I will focus in the leaf-aquatic ants and how the different habitats that they live could impact the bacteria community. Aphanogaster ants, which is our local ants collecting Westchester, collecting in Gordio Natural Area. And then finally, we'll focus on other 4 species of leaf-aquatic ants and how they're fungi, because those species, they eat fungi, and how is the bacteria on their fungi. So for Daxatone ants, take a look at this beautiful head. Look this huge mantle. So these groups of ants, they live in the south of America. So I collect them in Amazon when I was there. So the huge ones, it's pretty big like that. So I thought that they were very aggressive, and I was very surprised that they are not at all. I could let them walk in my arm and they didn't bite you. I was ready for a bite because dessert, right? I'm collecting them, I'm killing them. But no, they're kind of relaxed. Maybe it's the hot weather, right? I understand that. That's why I escaped here, right? So I don't like the hot weather. I like the winter, including so. But these groups of ants, we don't know much about that. We don't know what they're eating. We don't know, we think that they are predator because of the mandible, but they make their nest in the top of the trees in Amazon. So it's a challenge for you to collect them, right? Can you imagine find a colony of these? How many trees you need to climb and go? So my collaborators were there. They collect those by climbing those trees in Amazon, collect 2 big colonies. And then I had a chance to look the bacterial community associated with that. And for this paper specific, we want to understand how does the microbiome community differs between different colors, different sample types, if you collect entire ants, or just the castor, just the butt of the ant, or the different development case available in their colony, the different development stage. We also want to compare if those ants were collecting the wild directly or kept in the lab. That would change the bacterial community. And finally, if diet, we also have an impact and predict part of the bacterial community. So I know that this is a busy figure, so bear with me, we will go together what is it taking home here, okay? So this is a very common figure for microbiome study. So every column here, it's a different sample, and every color is a different bacteria. So when I got this data, and I want to emphasize, this is a big data, so I'm talking about genomics, it's a lot of material, just our human eyes cannot find patterns. So we need to use the stats and big data, genomics, tools, coding to get this data. But when I got this data, the first thing that even my human eyes were able to identify the pattern is like the diet that we are feeding them in the lab has a completely different colors compared to the other ones. And all the babies, all the larvae and pupa available in this colony was also pretty colorful compared to the other one, right? So I still need to use my stats to see if that pattern makes sense, but even my human eyes can say, yeah, something is happening here. So the next step then was showing like how it's the bacterial community associated, but now using stats. So this is the NMDS plot, meaning dots that are close to each other have similar bacterial community, dots that are apart from each other are different. And for this study, we are focused on different colonies, sample type kids, babies and workers like adults ants, comparing ants that were collected directly in the wild and do the DNA extraction process, or ants that was kept in the lab for a while, and also comparing the ants that was in the lab feeding with our specific, very sad, boring diet compared what it is available in the nature, in the Amazon. So when we get these results, we apply stats, and we saw that belong to different colony and have, or be a baby or an adult for those ants, we can use this to predict the bacterial community. However, not sample type, not different castas, not the lab or the wild sample. And that for me was shocking. Or even when we change the diet over a time in the lab, we don't see the difference. And especially for these 2 results, the lab and the wild, I think this was really important. And I was for a while like, what the heck there, right? Like how you were in Amazon, it's so different than the lab. I was in Chicago by that point, like very different in Chicago outside, right? So how the bacterial community are not changing here? And we are convinced now that these ants, they need those bacteria. So they use all their energy to protect their bacteria, because these bacteria probably are providing some important roles for the ants. So they cannot just lose those bacteria, otherwise they will die. So that's why we're not seeing difference with them. But that was a lot of crying process when I got the data, right? Like, well, what did we do something? Yeah, okay. So let's move now for the next study, and the next one I want to focus now in leaf-cutter ants. So this was conducted in the south of Brazil, in São Paulo, the state that I came from. And these ants, they are native for that region, but because we changed the agriculture and we put all their food available in the same place, like in a crop rotation, they don't need to go far away to find their food, so they can just like become huge colonies. So even if they are native, they actually become a really bad, like, economic importance for those crops, because they destroy everything, because they become huge colonies with other predators. So a lot of plantations there in Brazil, they use a heavy pesticides to kill those ants should reduce their size. And guess what? They kill everything, including ourselves, but not those ants, right? So they are pretty fine. Like exactly the same case of the fire ants here in the US. Kills everything besides the ants. However, in this study, we collect 75 different colonies of ants all over the state, and we compare the ants that was collecting the natural biomes and agriculture. In São Paulo, we also have another rainforest, but it is the Atlantic rainforest, not the Amazon. And we also have Cerrado, which is kind of a savanna. So we are trying to compare those two different biomes, which is natural, and also how those ants that lives in the plantation, like eucalyptus, citrus, and see how those have impact on the bacteria community. So for this study, we are testing the different habitats, natural or agricultural habitats, and really physically the geography of the São Paulo state, and see how these could help us predict the bacteria community associated with those ants. And we found out that citrus has a unique, ants that was around orange, has a unique bacteria community associated with them, but not the other ones. And that's pretty interesting because citrus is one of the things that Brazil sells outside, including to here, US. So a lot of our juice, orange juice came from there. And guess what? We have an odd bacteria community there, Tucamorella. And we don't know how the Tucamorella is there, but it is there. And that probably could be because from all these other crops, the citrus is the crops that we have less law regulations in terms of what kind of pesticides they put in their plantations compared to eucalyptus and sugarcane. So that could be now a potential problem for understanding how that will impact the economy, if it is impact the economy. But now, my former advisor and our collaborators will let us to go collect those ants. We are trying to come back and try to investigate how those Tucamorella are more associated with citrus compared to the other ones. So that could be interesting, so stay tuned. The other important aspect for this study was that we were able to also see the different geographic location could impact the bacteria community associated with those hosts. There's a follow-up on this study, and that will be in few slides when I show you the study that my student, Alex Giannaris, are conducting here in Westchester. So stay tuned. Let's talk about our local girl. That is a Phenolgaster ants. In ant colony, we just have humans, right? That's why I'm talking local girl. So we have tons of those Phenolgaster ants in the basement of the biology because we are now collect them in the Gordon Natural and bring them inside to know more about them. So thanks to several guys here, in the folks in the audience, we are now understanding a little bit more about the bacteria community associated with those ants. But more important, those ants, they are really important for these local ecosystems because they are seedless person. So if we lose these ants, we will have huge important impact in the ecosystem around us. So let's know more about them. So Lily, my grad student who's about to graduate, just published this paper last week, and she was the first one who got a chance to look the two different species of Phenolgaster that we have available here in Westchester, Gordon Natural Areas, Phenolgaster fulva and Phenolgaster rudis, and she did found that there is a difference between the bacteria communities between these two species. The next slide, we're now really thinking how the different development stage available in this colony, if these different development stage have an impact on the different bacteria community. And a huge chunk of you are seeing here, it's the eggs and the larvae. They all have a different bacteria community, similar to what I did with Daceton, have a different bacteria community compared to adult ants. So both belong to a species and different development stage can impact the diversity of Phenolgaster. I'm not going to much detail because the next, so Lily's graduating soon, so she will give a talk, I can invite you all. But the second chapter of Lily's thesis is look, once we increase the temperature, how the climate change mimic what's supposed to be the climate change in 100 years, how that will impact, or it will impact the microbiome of those local ants, if they will lose some important bacteria that help them survive in those woods outside. So stay tuned, Lily will come talk, but the answer is yes. Okay. So the next one I want to show now, that's the follow-up of the Lificara ant study. And that was the Alex Janaris study, our undergrad in biology department. So Alex already submitted her paper, and she's now submitting another chapter that she was invited to talk about the ants from Brazil. And since she studied ants from Brazil, she is now qualified to help us to write that. But Alex wants to look now, instead of to look just one Lificara ants, she was able to see 4 different species of Lificara ants, and she do find, and if you look all of the species in evolution of these Lificara ants in Brazil, they're all more in that part of the clade compared to the other ones. And she did find that there is a difference between all the different species of ants, but not just the ants, also look at the different fungi, compare ants that was collected directly in the wild or ants that was bring to the lab microbiome, and also ants colonies that was added to the treatment of pesticides to see how all of those factors of different categories treatments could be impacting the bacteria community associated with those ants. And Alex did find that there is different species could also help us predict the bacteria community associated with the Lificara ants. But especially who is driving more that different is the Atta levigatum and Atta sexton. So those two have quite different bacteria community associated with them. So stay tuned for Alex explaining you more how she's presenting a talk. She's presenting her poster in the research day. She's also presenting a talk in Ecological Society of meetings that is happening in Longwood Garden in April. So stay tuned. But another result of Alex that I want to show you is that the fungi for those ants are actually the diet, is their food. So here is the ant and here's the fungi associated. So the ants cut the leaves to feed the fungi, and then the ants eat the fungi. So, and then when we look at the bacteria community in those fungi and compare to those ants, we still find difference between both of them. So stay tuned, she can talk to you more about why this is happening, which bacteria that they have in difference. But I hope that now you can see that there is a difference for our original question, that we do find that ecology are important for Dacetum, Aphanogaster, and Lificara ants. So now let's talk about phylogeny. I'm going to check my time because I can speak here for hours. Okay, so when I focus on phylogeny and see how that's impact, the idea is I will use the most robust phylogeny available for the group of ants, and I can compare with those data that I've been showing you, the microbiome data, and see if both of them are co-evolving or not, or if they co-diversifying or not. So the way that I did this for the spiny ants, groups of ants that happen in Australia, Asia, and Africa, and we did find that both bacteria and the ants are co-evolving. What I want to do now is applying this for turtle ants, which is the cephaloid ants. So those ants happen from the south of US into the end of America continent. So they happen all over the place. And these groups of ants are known already to have this established relationship with several bacteria for more than 50 million years old. So one of my friends and collaborators were able to look 25 different species of turtle ants, cephaloids, and they did find that there is a core community of bacteria that it's always there with them. And they also did find that these bacteria communities are co-diversifying with these 25 species. So what I want to do next is increase by a lot. There is about 100 species of turtle ants in the world. So now we got 3 times more turtle ants species. And we want to see if what is the ecological factors that help us to understand more about the bacteria community associated with them. We now can assess both the bacteria and the eukaryotic communities like the fungi and the nematodes, and see if both of them are interacting with each other yet inside of those ants. And finally, if we want to see if those now increasing by a lot, if those ants still have a core community associated with them, if they are also co-diversified. So the first thing that we were able to see is that all those ants have a core community. So meaning that doesn't matter where those ants were collected. If that was for the key, Texas, Amazon, or south of Argentina, those ants have always the same kind of bacteria, the same lineage of bacteria are there, which is kind of impressive how those ants are able to do that, right? Think about it. And then when we look these you know how to identify this. Every column, it's a sample. Every color, it's a different bacteria community. So when we get all our results, and I compare with these with previous studies in the past, I was able to see that those groups of ants, those girls, they had a very remarkable conservative microbiome. So this is a field where the technology is changing all the time. So the way that those studies generate the data and the way that they analyze those data in 10 years ago completely changed the way that I do things now. So it's always update, always change, which is also part of the challenge, but it's also pretty good, pretty fun. That's how, if you work with genomics, that's what you're supposed to do. And comparing my fresh new results with the most updated way to analyze them, you still come back with exactly the same kind of bacteria that was showing up. Doesn't matter if those ants are from Florida, King, Amazon, or Savannah in Brazil, which is quite impressive. Okay, so how do those ants get that? That's still part of the question, but I will have some ideas. And belong to a species and the different biogeographical locations help us to explain part of the bacteria community associated with that, which we expect. But now that we have way more samples, we can actually see that that sounds like the pattern. The cool thing about the technology now that is always advancing, we can now look all the eukaryotic microbes and also the bacteria and see if they are interacting with each other inside of the ants. So what you're seeing here, I have here just the core community, just the bacteria community associated with those ants, and here all the 18S, all the fungi, nematodes associated with those ants. And all those lines, the red line, this is our correlation happening between them. So the blue line, it is the negative correlation. The red line, it is the positive correlation between them. We still don't know if this is happening as a predation, it's a symbiosis, it's a mutualistic one. But the fact that now we have the technology to see different kingdoms interacting with each other inside of a single insect, ants, it's pretty remarkable. And I can probably spend the rest of my career, and maybe the rest of the career of my students, just to design experiments to understand more the interaction between those kingdoms just from that image. Pretty cool. Okay. Finally, let's see if both of these bacteria and phylogeny are bacteria and ants are co-evolving. So I used the most updated phylogeny for these groups of ants, the turtle ants that was published in 2022. And I had a chance, I was part of that lab at that point, and I used exactly those same samples that generate the most robust phylogeny to analyze and generate these microbiome data. So it's exactly the same sample. And then I applied the mantle test, the PACO test, and I visualized all of this with a tandem run because I was trying to have a very conservative vision to see if the co-diversification, the evolution of those of them are happening or not. So when I look at my data here, so the way that we will identify this is the host, will be the bacterial lineage. If you see those lines like straight forward, that means the co-diversification is happening. If you start to see some tangle, so that means the co-diversification is not happening. But I want to emphasize that our human eyes cannot handle all those patterns, right? So we need to apply some stats, and that's why we are very conservative for that. So we just rely on results that have both the PACO and the mantle test exactly the same. So again, I know that this is a busy figure, but everything that you're seeing in purple was significant. Everything that you're seeing in yellow, it is not significant. And now we are separating all the big lineage of bacteria associated with those ants because we want to make sure that every lineage of bacteria can have a different history, evolution history with those hosts. So when we separated them, we saw that Burkholderia is the only one that we are seeing that are co-diversifying with the host with million years old. However, try to take a look here and use your human eyes to see if you can identify a pattern. It's impossible, right? So that's why we need stats here. And both were very confident that co-diversification is happening. To the flip side, the other two that come up with the same results was Wolbachia and Acetobacteriaceae. Then now we have evidence that it's not co-diversifying with the host. All this other one, it's kind of like still a question, and probably we can come back and design different experimental to target it, using different technology to target it specifically. strains of bacteria, the genome, to know if they are co-diversified with the host channel. But at least now we have some clues where should we start to investigate them. Okay, so for phylogeny, I hope that you can see that for Polyrex, that yes, but for turtle ants, yeah, that's depends, because now we know that we should look those lineages separately. Not for Bourbactia, not for Acetobacteriaceae, but yes, for Burkholdeриaceae. They are definitely co-diversifying with hosts. Okay, I want to use my last minutes to talk about diversity and inclusion. So why should we talk about women in science, parent science, or underrepresented in science? Is that enough? Well, a famous scientist, women scientist who still talk about women in science out there in conference, was approached by one of other faculty saying that, are you still talking about this because you're not talking, you don't have enough papers? And this scientist is very famous in the field, have more than 200 papers published. So even if those big senior famous scientists are still being approached by those scientists saying that those kind of things, yeah, that's enough evidence that we still should talk about this. Another example for a focus group study insects was this, that was circulating the Twitter, like insect pest management, where released like the advertisement of this conference showing just male scientists. That would be a keynote speaker. So this is not acceptable, and that's the reason that we still should talk about diversity and inclusion in science. So even if you don't care about this, but you care about the quality of the work, the science, so several studies have been demonstrating that groups diverse can actually solve a problem faster and with better quality. So even if you would just care about the quality of science that we are producing, we should invest on that. For the humans perspective, the women in science perspective, when we look, when we are talking about representation of women, it's normally that it's possible that you see this metaphor of the leak pipeline. So what do I mean by that? It is every time that the women advance in their career, there is a leak in the pipeline and less women will be available in that stage of their career. So women start pretty much the same if you were still master students, but in the PhD, there is a leak. So there's less women pursuing PhD, even less to become postdoc and full professors, way worse. Try to think about deans available in college or stage of the scientists in the final stage of their career who get more, let's not even talk about salaries here, right? But the causes why women are leaking in the pipeline to not become a senior in their field are plenty, including maternal leave problems or friendly environmental in science. So with that in mind, I invited other two women scientists to work with me and try to understand how is the representation of scientists who study ants all over the world. And then just by the females, I told you that the ants are more females, more inside of the colony. Just by the title, you can see our results is Spider Press. So in myrmecology, the majority of female only in the colony, because when you look at the scientists. So what do we did that? How do we do that? So we look for more than 5,000 of paper published since 19th. Before that, it's quite tricky to find information about the scientists who publish or find those resources available online. That's why we stopped in 90s. So when we look all those papers, we saw that women, we first look at two different authorships in the paper. The first author as a proxy in the beginning of their career, that's the students or postdocs. And then the last author is normally like the PI, the professor, the senior responsible for that paper. So in both these locations in the paper, women sign for less paper than men in the field. And the tricky, which was very surprising, is even when you plot these over the years, you can see the first author, the red, has a little bit of increase over there, but that's not enough significant when you compare with the median, with the average. And the worst part is the last author. So that for me, like there's way more fluctuation and no change over the 30 years. So I told you that I have 30 more years of my career, and that's now I'm working hard to change that line to be significant difference and to make that difference. Because what is happening here, it is even that women, we are attracting more students, those students are not finding a job and establish their career in the myrmecology world. That's a big problem. So besides to identify those problems, we also make several suggestions for future scientists, institutions, and journals, and for all the majority of the science men, for how you can overcome this. And I don't have time to go through all of that, but I wanna get your attention to the last one for all the men. If you are the majority of science, we need to come with you as an ally to help us to change those trends and translate this to better science for everyone. So stop to tolerate it and accept them behaviors from other men in public and in private. We need you as ally. And other study that was kind of conducted exactly the same time, also in pharmacology, for my friend, totally random, that we discovered in a conference, in a breakfast, that she was also working in a data set similar, looking at collaborators, they also look to the majority of the representation of females scientists in the ants all over the world, but they investigated this with a different way. So they look how the influence of the GPD of the country support doesn't have much impact how rich a country is, comparing to how the culturally those countries support women's engaging in STEM careers, which is kind of like challenge. And Lucky and collaborators were able to see the papers someone who published just once and never published again. And then for those in academia, they saw that majority of the women signed up for those papers who published once and then leave academia. So that's a problem of retention, because if you think like you have undergrad, black masters, PhD, postdoc, it's so many years that we are focused in training, right? The next, yeah. And then those people are leaving academia before like they get there. So that is a huge problem that we as myrmecologists, as scientists, now are thinking how we can overcome this. Okay. I will try to speak really fast. Sorry, guys. So I'm changing gears, but not so much. And for that one's like, I'm talking about now parenting science. So several studies have been demonstrating that become a parent has a huge impact in how efficient you can conduct your work, and scientists are one of them. So, and that study was, there are several studies, but my favorite one was conducted by this group, which is also a Brazilian group called Parent Science. And they put this graph, which is show like how is the publication rate affect after the arrival of the children. And when that happened, I was here for my children. I was like, oh my God, it's still going down. Like they say that the women start to recover after the third year to come back to increase their productivity again. So that hit me hard. And with that, I invite other moms in the myrmecology to see how was they doing. And I invited the entire myrmecology community to actually answer a questionnaire and see how did they get when their kids. And then the idea is we still can publish papers, we still can work in our lab, but how those other scientists who need to go to the Amazon, collect ants, do this when you have little kids in your house, right? So that was my main struggle for that moment. So we invite scientists with kids and no kids to compare. And we do find that for regular academics in a research routines, meaning you don't need to travel, there is no difference between number of publication, proposals submitted, numbers of mentees and classes that they taught over 5 years of their arrival with children. However, when we look for activities that ask for people leave their routine, their niche where they have all the help that they need, for example, go collect in Amazon, field trips, public speaking, or attend a conference, that we still find difference after 5 years of their arrival of the children. So, and we start to look at 5. So when Nikki turns 10, my daughter turns 10, I can come back and ask if is that still a trend after 10 years, because I'm living this just with one observation and still how hard it is to leave to go to a conference, right? Or to go to Amazon, collect money kids. And this could be one of the potential causes why we still have so many people leaving in the college field and finding probably better jobs paying for sure, but still losing a lot of years training scientists to become scientists. So we still need to think about a better way as a community to overcome this. Another study that was conducted by my student here, Ethan, which is about to graduate. Okay, I will leave this one because this is better, Ethan. Have your name. So was starting from this idea that our identity shapes our perspective. We invite again the rheumatology community to answer another online questionnaire with scientists spread all over the world to think about the different terminology that we think it could be problematic, how these affect you based on your background. And we have some really bad terminologies. Italy is looking more than 13 different terminologies that was suggested by those survey. And we saw even with a very problematic term like slave mating in, just 40% of the scientists think that we should change that. And that is definitely driven by the fact that we have way more male white scientists in US and Europe actually that want that change. So even in this 40%, we have awesome suggestions. Instead of to use this late making ends, we are asking the entire rheumatology community to suggest new terminology that will be less harmful for the entire diversity community of ant scientists all over the world. So turn out that some of the radar could be one of the substitution for the one, and we are now in the phase. So Ida will be presenting these results in research day as well, in the Ecological Society of meetings as well. And if you want to know more, come talk with him. But that we are writing a paper to make those suggestions with scientists all over. So I want to thank you, especially Manu Lab is turning 3 years old. We are a great diversity group of people who do a lot of tons of outreach. We also do a lot of film collections all over the world. We present papers, we have a lot of fun, we always have snacks, and if you want to come back to Marin 13 snacks, we have a lot of parties, so we have a lot of parties, a lot of ant antennas pictures, and I want to thank you all of them, especially because they're incredible, they make my day better every day. And with that, I also want to thank you all for being here. Sorry that I speak a little bit more than was ideal. So Westchester, thank you so much. Thank you, my former two labs, and now I have my current lab here. And I also want to thank Nikki, my inspiration, also day by day. And thank you so many collaborators all over the world and the staff for funding all the women science that always support my careers. And if you have any questions now or in the future, please come talk with me. I'm in Marin 313, but also here's the website, if you use my email. So thank you guys.
Time, we want to point out some students might
Yeah.
have their class, so
Yeah.
they have to go and
Yeah.
for sure, Dr. Ramalho.
Absolutely.
Take some
Go to your class. Yeah. But if you have any questions, you know where you can find me now. Also, if you find good wins.
I saw my friend Erin in your.
Yeah, Erin is from Marriage Lab, but we are like cousins. So we are.
He showed me the roaches once.
Yes, they are now in Marriage Lab, so it's cool. But they were living in my lab for a while.
Congrats.
Thank
All right. I think I'll stop the Zoom here unless you guys have a question. Well, you have a question?
Thank you.
Oh, I have a question.
Oh, wait, I can't hear you. 1 second. My computer is being goofy. Let me,
Can I turn off the volume or that will be not good?
let's see. Can you try talking again while I'm outside?
Yeah, I have a question. Can you hear me?
I don't know.
Yeah.
So is there a continuity between the microbiomes of ants and termites, whether phylogenetically, ecologically, compositionally, whatever?
Excellent question. I think no one, I'm hearing my echo. Are you guys here? You hear me, Ross?
Yes, I can.
I think it's okay.
Okay. So I think there's no one study that put both on in the same. But what do I know about termites? They have a lot of proteins in their gut that help them assess the digestive cellulose that they need. I know that the termites also have Bulgarkia, which is very important in bacteria famous that ants also have it. So there is for sure some overlaps of what do we know from bacteria that lives in their termites and then lives on ants, but there's no just one strain. Another thing on that, once I did a meta-analysis where I look for what what do we know for the termites microbiome perspective? And I found just like, I think 8 studies, and there was a study in termites, microbes all over the world using the technology available today, next generation sequence. So I have tons of friends that works with termites, and I was like, what? Let's do that. Exactly what I actually should answer for. But it's not much study yet in terms of with the technology available today compared to the ants work.
Mhm.
Yeah. So maybe we should.
In another lifetime. Yeah.
Yeah. John?
Very nice talk. What do you think the function or benefits perhaps of the Burkholder ACA family members might be? They seem to be pretty prevalent there.
Yeah. So another collaborator of mine, they look 4 different strains of Burkholderia, and not all the Burkholderia were co-diversified with others. Some of the lineage of some strains of Burkholderia was, and for these 4, they found evidence that they are upgraded nutritionally for those things. But it's through the technology, they use the shotgun metagenomics, so they can have not just one gene, like I was looking, the 16S, they are looking actually all of the entire genome to know what kind of proteins those bacteria are producing. Yeah,
So it's nutritional.
nutritional. Yeah.
Have any studies that you're aware of been done in which they seek to modify the composition of the microbiome, perhaps through either genetic means or alternatively through the use of antibiotics?
Yes, we are doing this now with the Fenogasther ants in the basement in Westchester. So first we start answering questions like, okay, how we can explain the diversity first? What it is out there, right? Like the species determining its development stage, its diet, and then we move to the next step. Okay, what about now antibiotic? If you will add the antibiotic, how is the effect on the phenotypic? What is the variation that we can see? How that changed the fitness of those ants? Do they become healthy adults? Do they not? So we are answering these and mimic with the morphology and development and behavior together with the microbiome kind of analysis to see which one had impact. Lily, you were part of her committee, so we will see in a few weeks that Lily was also able to increase the temperature and keep those colonies in the temperatures. In microbiome world, for the climate change with people who study ants, they normally do the study. They get one ant and increase the temperatures and mark down when they die, when they die. But you need to think ants live in a colony. So the colony is also considering a superorganism, and they use collectively behavior to actually reduce the temperatures or to how overcome those temperatures. So what we did, no one for a long time was thinking that climate change would affect ants because of that. So a recent study was published in 2023 that actually demonstrated that that's not quite sure. There is a limit that the ant colony can handle. After that, everything goes down. And then when I saw that paper, I was like, okay, what about look this through the lens of the microbiome perspective? If you increase the temperature in the way that will be, how these will impact, first, will impact the bacterial immunity. The ants will figure out a way, like use all energy to keep the bacteria, the good bacteria that they need to survive, or not, or they will collapse. And Lily, the results is demonstrating that they collapse. They lose tons of whole bacteria, which is somehow important for those ants. And yet, how do we know what is the real function for those bacteria? It is using the shotgun metagenomics to actually assess the entire genomes of those bacteria. But we are still seeing that those changes impact the bacterial immunity. I think the next level is transcriptomic or full genomic of those bacteria.
So if they disrupt the Wolbachia, yet the ants survive, will it affect their
The ants don't
reproduction?
survive.
Oh, they don't.
They don't. They stop to produce. One of the first change that we saw, mortality increase, and they stop to produce new babies, new larvae and pupa. And then eventually they all die. Yeah, and we kept the colony 5 weeks in 32 Celsius. And then we saw a peak of mortality, so we stopped the experimental when we saw that to collect the different development stage available to see how that impact. But we removed those colonies from the 32 to see if they can overcome, because the queen was still alive. They could not collapse. Everyone died. So it's not, there's no plasticity on those, like to try to recover. Yeah. Pretty sad, which is huge impactful for important local ants, right? Who is a seed disperser. Yeah, so that will be the first paper published demonstrating that climate change could be impacting bacteria community for important local ants. So we are pretty excited, yeah. Any other questions? I didn't see the chat if we have questions. I don't think so. Okay, we'll check. Just say thanks. Awesome. Thank you. Yeah.
Thank you
Thanks.
so much.
Thank you. But I thought that I speak more than what you.
No, it's so interesting.
It's very interesting.
Thank you.
Are you sending